Hi everyone, thank you for listening to this presentation. My name is Sarah Musavi. I'll be presenting a semantic clustering algorithm for images of human decomposition called SChISM. Nowadays, large image collections are everywhere. They drive research and facilitate analysis in many domains. However, there are specific domains that the type of data they work with is not necessarily curated or in research-ready format. One example is forensic anthropology. In this case, we work with 1 million images of 500 subjects collected over 8 years to study human decomposition. What's specific about this dataset is that the content of the images evolve over time, as it's shown in this example. For such datasets to be used by researchers, we need to be able to extract relevant information from them. That means we have to have a prior knowledge of what each image holds. This knowledge can be added to the data through a manual labeling process, which is very expensive and time-consuming, or through an automated process. Clustering is an unsupervised way for gathering information that are relevant together from an uncurated dataset. Traditional clustering methods, however, are not capable of extracting image features well. Deep learning-based methods are also not the best fit when it comes to evolving content. So we propose a sequence-based clustering that leverages the underlying structure of the dataset and tracks feature variations over time. As a result, we would allow exploring data based on the content of those images. So one would be able to study a body part from fresh to skeletonization. The algorithm of SChISM is as follows. We first map the images to feature representations and then use a window-based sequencing algorithm to generate short snippets of similar images. Finally, we merge these short snippets to form larger clusters. The algorithm for window-based sequencing is that we compare images within a window, and based on the level of similarity, we decide if they should be assigned to the same sequence or not. In order to evaluate our method, we use purity. We also define 3 new metrics that are suitable for evolving data, namely gap, essential cluster, and inclusion. Gap is defined as the number of missing time steps for a given class. In this case, it's 3. Essential clusters are the clusters that their presence result in capturing maximum number of time steps for a given class. For this example, C2, cluster number 2, is not an essential cluster because those time steps are captured by cluster number 1 as well. So the number of clusters, essential clusters, here is 2. Inclusion is defined as the total number of time steps captured by the essential clusters for a given class. We evaluated SChISM by comparing it with a pre-trained transfer learning-based CNN, as well as the state-of-the-art for unsupervised representation learning called deep cluster. The result of our evaluation for purity, gap, essential cluster, and inclusion for the pre-trained CNN, deep cluster, and SChISM is shown in this slide. As you can see, SChISM generates highest purity for the clusters, minimum amount of gap, and more consecutive clusters, whereas it generates a smaller number of essential clusters while it captures maximum number of time steps for a given class. With that, I'd like to thank everyone. Please contact me if you have any questions.